In today's lecture, we'll start with the topic glycolysis. So, we all know that to do every small thing, whether it be like moving from one place to another, or like running, or simply activity like speaking or writing, we require energy. But the question is, from where does our body produce this energy and in what form? So, to answer this, let's imagine a car running in a racetrack. Now, if I ask you, how is the car able to run in the track, you will say it has fuel inside it, right? So it provides engine the energy to run. Like so, similarly, in our body also, the cell inside the cell, glucose is like fuel, and glycolysis is the engine that provide the energy in the form of ATP so that the cell could function. And how is this ATP produced? That can be the question. But before that, the question could be, from where does we get glucose? So we can say that glucose comes from carbohydrate, and carbohydrate is like a thing that we take in our diet. Like we eat roti. Roti has carbohydrate. So and glucose is the part component of carbohydrate. So in that form, that we take carbohydrate in our diet and glucose enters our body. Now, try to relate this. Our body is glucose fuel, glycolysis engine that produce ATP, which is actually the energy to speak, write, etc. Right? So it provides us energy to speak and write. So this is our basic understanding of glycolysis. Now we'll try to understand what actually happens inside our body. So glycolysis is a Greek word which means glycos, sugar, lysis means splitting, which is totally stands for splitting of glucose molecules. So we can say glycolysis is a process in which glucose is broken down into pyruvate in 10 simple steps, and it was given by Kostev, Amden, Otto Meerhoff, and J. Prance. Okay, now we'll see the steps. Before that, we'll get to know that it's an anaerobic process, right? Very key step, very key points. It's an anaerobic process that does not require O2. And where does it take place? In the cytoplasm of the cell. This is a very detailed version of the cycle, which appears to be very complex, but we'll try to simplify it. So the glucose first breaks down into glucose 6-phosphate in the presence of hexokinase. And in this step, ATP is consumed, energy is being consumed. Okay, now in the second step, this glucose 6-phosphate in the presence of the enzyme glucose phosphate isomerase break down into fructose 6-phosphate. It might be appearing very dangerous to us in the first step, but if we'll try to read it again, it will actually make sense to us, right? So, wherever there is consumption of energy, the enzyme will end with ase, kinase. Similarly, in this step also, the fructose 6-phosphate is being broken down into fructose 1,6-bisphosphate and an ATP is consumed. So the enzyme here is phosphofructokinase. Now, in the first step, we remembered the enzyme with a very simple step that wherever the energy is consumed, the enzyme ends with kinase. But in the second step, we can remember it like as whatever is the like substrate of the next product. The enzyme name would start with that glucose phosphate, and because here it is forming in the getting converted from glucose to fructose, so the enzyme name is ends with isomerase. Again, fructo. So we have phosphofructo, and because the ATP is being consumed, so the enzyme is kinase. Now, fructose 1,6-bisphosphate broke down in the presence of aldolase in dihydroxygen phosphate and glyceraldehyde 3-phosphate. Now, both the these products will undergo same breakdown, and the same enzyme will be taking part here. There is no change. But here, the inverted arrow means implies that they can interconvert, like it can convert into this and this can convert into this, right? Similarly, here, the enzyme responsible for this step is trisophosphate isomerase, glyceraldehyde 3-phosphate 1. Now, in the first initial step, we saw that ATP is being consumed, but now from here, everything will be produced. So glyceraldehyde 3-phosphate, when broke down into 1,3-bisphosphoglycerate, NADH is produced. Same 1,3-bisphosphoglycerate, when broken down into 3-phosphoglycerate, ATP is produced. Same 3-phosphoglycerate broke down into 2-phosphoglycerate. When 2-phosphoglycerate broke down into phosphoenol pyruvate, we have one water molecule release. And when phosphoenol pyruvate break down from phosphoenol pyruvate to pyruvate, we have ATP produced. So what we saw, one, two NADPH molecules being produced, four ATP molecules being produced, and two water molecules being produced. So what's the uh, tricky part to remember this? So we'll give a simple trick for this. Good girlfriends will class daily by 3 PG, 2 PG, play ping pong. Is it easy now? Right. So, in the process, we see glucose break down into two molecules of pyruvate, two molecules of NADH, and four ATP are produced. But there is a twist. The net gain is of 2 ATP. Why is it so? Not 4 ATP. So, think of it as a vegetable vendor. You are a vegetable vendor who invested 2 rupees on ingredient and sold delicious cuisines at a total price of 4 rupees. But what is the difference? You invested 2 rupees, so the net profit is of 2 rupees. So similarly, in the first steps, you invested 2 ATPs. Now, therefore, the net gain is of 2 ATP, not 4 ATP, right? This is a very key step to remember. That's why in glycolysis, one molecule of glucose provides 2 ATP as a direct source of energy, and NADH and pyruvate as energy vouchers. They are energy vouchers, like NADH and the pyruvate produced are energy vouchers that will be later used in the process, right? So with this, we wind up our topic glycolysis. Thank you.